A genome is the collection of genetic information present inside the cell of an organism. Humans have genomes, as do dogs, flies, strawberry plants, and any other living organism. The genetic information in the genome is almost completely identical between members of the same species. There's a little bit of genetic difference, called DNA variation, which contributes to different traits being present among that species. This segment talks about the relationship between DNA variation and traits, using an analogy for something we're all quite familiar with, a recipe for chocolate cake. All right, so let's talk about the human genome, so the collection of all the genetic information inside each of our cells. The Human Genome Project from the 90s through the early 2000s was geared at identifying all the A's, T's, C's, and G's in the DNA of our cells and identifying the number of genes we had and where those genes are and beginning to learn about them. And we learned some really interesting things along the way. Only about 1 to 2% of human DNA actually is genes. So there are 3.2 billion letters, A's, T's, C's, and G's, and only about 2% of those actually are instructions. So what's the rest of it? Well, we used to say that it was junk. That is not a word that we use anymore. It is not junk. It actually looks like a large part of it's probably regulatory that controls when certain genes are activated and information is made and when certain genes are silenced. In other words, when protein gets produced from the genes and when there's no protein from that gene being made. The genomes of any two humans differ by less than 1%. I love this. This means that we are much, much, much more identical than we are different. Less than 1% of our DNA is different between any two people, no matter where on the planet I pick them from and compare their genetic information. The changes that do exist, and there are a lot of them, some of them are a single letter long, like I told you about with the sickle cell anemia, with that 63,000 hemoglobin beta gene, one letter change leads to sickle cell. Some of them are thousands of letters long. So there's a lot of change between any two people, even though we are very, very similar. So that begs the question, what happens when the DNA changes? And so for that, we're going to use an analogy. I'm going to assume that everyone in here either likes to cook or likes to eat what people cook, or just likes to eat. So that should cover us. So here's the analogy that I want you to remember. A cookbook is a collection of lots of different recipes. And these recipes have they tell you how to make lots of different things. And when you follow the recipe, you make that product, whether it's chili, pork loin, or red velvet cupcakes. That same analogy holds for our genome. Our genome is like the recipe book. It's the collection of all the genes. Now, there's a bunch of non-recipe information in our genome. I only told you 2% of our genome is recipes. So there's a whole bunch of stuff about how to fold the napkins and what wine to choose and how to set the table. But there are about 21,000 recipes, genes, in our genome, and they each make specific products. Everybody with me on this analogy? Okay, good. So here's a recipe for chocolate cake. And you can see it uses sugar, flour, cocoa powder, baking powder, etc. all the way through. Let's start to change the recipe and see what happens. So if I have brand A flour and I shift it to brand B, will my recipe, will my product change? Well, so my grandmother would tell you that if you use anything but white lily, you're in trouble. But in general, this is a silent change. This still is a chocolate cake. It still tastes like a chocolate cake. So that's a silent recipe change. Strawberry frosting. Still a cake? Yeah, a different cake. Some of us might like strawberry over chocolate better, but it still meets the definition of a cake. Okay, let's go back up here to the flour. No flour. Is this a cake? I know you can make a flourless cake, but bear with me a second. we now have introduced a pretty significant change so that this is no longer recognizable as what the recipe is intending to make. And we can have lots of other changes. I can add a little bit too much milk and a little too little flour, and I end up with something that's kind of soupy. So there are lots of ways that I can change the recipe. Some of them are completely silent, some of them make a huge mess, and some of them lie somewhere in the middle. I want us to take this exact same analogy to our genetic book of life, to our DNA, our genome. There are some changes in our DNA that are completely silent. These happen all the time. In fact, most of the change, most of the differences in your DNA between you and me, or between you and someone from Alaska or South Africa or Tanzania, most of these changes are silent. No impact at all. Some of these changes are what are called polymorphisms. Polymorphism means many form. So these are changes like chocolate to strawberry icing. They don't change the final product. It still is a recognizable protein. It still is a recognizable cake, but it's slightly different. The DNA changes that lead to eye color are perfect examples of polymorphisms. And then there are genetic changes that alter the function of the protein in such a way that it no longer can do what it is meant to do. And there are consequences of that. And we're going to call these mutations. Now, Mrs. Adams, I know as a biology teacher, the official definition of a mutation is any change in DNA. So I recognize that from the strict biological sense, any change in DNA is a mutation. I'm going to go with the clinical definition, which is any change that impairs function in some way. So I beg your forgiveness for that. So these are the ones that lead to disease. These are the ones that you may be familiar with. Now here's the challenge. In one person's eyes, a genetic change may be a mutation. In another person's eyes, it may be a polymorphism. Individuals that have certain forms of deafness caused by changes in their DNA do not consider themselves diseased. They are part of a thriving community, they have a vibrant language, and they just fall somewhere on the spectrum of the amount of hearing. Individuals that have certain forms of short-limb dwarfism would not necessarily consider themselves to be diseased. Others would say they have a disease and they need to be treated. So this spectrum between polymorphisms and disease is very tricky and very difficult to tease apart. Most changes in DNA are commonly due to errors in DNA replication. So every time you want to divide your cells, you have to make a complete new copy of your DNA. All 3.2 billion letters have to be copied. That would take a really long time for you and I to write out by hand all 3.2 billion letters, and there would probably be some mistakes. And the systems in your cells that copy that DNA do make mistakes. Often they get repaired and you never even know the mistake was there, but sometimes the mistake stands, and the new cell now carries that change, and every cell that comes from that carries that change. The other place we often get DNA mutation is from DNA damage, like exposure to ultraviolet light, exposure to asbestos, exposure to certain carcinogens in cigarette smoke, for example. These damage your DNA, and oftentimes your body can repair them, but in some cases that repair gets missed, and now every cell that comes from that carries that mutation. Now, will that get passed on to the next generation? Depends on where the mutation occurs. If the mutation occurs in a body cell, in a skin cell, I may have all the skin cells around it that divide that carry that mutation, but I'm not going to pass that on to my children because that mutation didn't occur in the egg or the sperm. If the mutation occurs in the egg or the sperm, and that egg or that sperm is part of a fertilization event, then that child, every cell in their body, will now have the mutation on the copy of that chromosome that came from me. So if I have a mutation in a sperm cell that gets passed on to my son has a mutation in every single one of his cells. He will then pass it potentially on to his children, who have the potential to pass it on to their children. So things pass through families because somewhere way back in the family tree, or maybe not so way back in the family tree, a mutation occurred in that gene, and it occurred in an egg or a sperm cell and got passed on and now showed up in all the cells of an individual. So that's why when we say some mutations are inherited, versus mutations that occur in cells like your skin or your liver that don't get passed on to the next generation. There was a question. Yes, ma'am?
What's an example of something that would be changed and then passed on?
Something that would be changed and passed on. Sickle cell anemia, for example, in some cases shows up in a family that has no history of sickle cell because there was a brand new mutation in an egg or a sperm of a parent. And that change disrupts the way that the red blood cells function. Any genetic disease that you can think of at some point in history was a brand new mutation. Started as a new mutation and then slowly made its way through the population. Now, on the flip side, cancer is an example of mutations in cells that are body cells that don't get passed on. And next week, we'll talk about how changes in our cells lead us from normal growth to abnormal cancerous growth.
Since these errors can occur with DNA replication, which is dividing the cells, how many times, it's kind of a crapshoot, how many times do your cells divide in a lifetime, and can you do anything to inhibit that?
Well, you can stay away from things that damage your cells. You can try to boost your repair systems with antioxidants. Now, that does not mean you need to go out and take bottles and bottles of antioxidants, but antioxidants help your DNA repair systems function. It's estimated that every one of us has 30 to 60 brand new mutations, half of which came in dad's sperm and half of which came in mom's egg, that were not present in either parent, but happened specifically in that egg or sperm. Most of those are completely silent. They're like swans down to white lily flower. You never even know them. But you can do things to try to stay away from things that you know damage your DNA and try to boost the efficiency of your repair systems.
How many times do your cells divide?
Well, you start out with a single cell, with a fertilized egg, and there's about 100 trillion cells in the adult body. And many of those cells, like the cells in your digestive system, divide on like a weekly basis. So I can't give you a good solid answer for that other than a whole lot. All right, let me move us on. I know we've got some questions, but let me get through some of these other pieces. Change can occur at lots of different levels. Woo! You can have entire extra or missing chromosomes. If you know an individual who's impacted by Down syndrome, every cell in their body has an extra copy of chromosome 21. So instead of 2 copies, one from mom and one from dad, they have 3 copies of chromosome 21. And that extra copy is what leads to all of the clinical symptoms associated with Down syndrome. That's genetic change on a really big level, having an extra chromosome. At the opposite end of the spectrum is having genetic change on a single letter, where one single letter of those 3.2 billion is different. And you can actually use this to identify identity and ancestry, so it's used in forensics to identify if a sample from a suspect matches the sample left at the scene of a crime. It's used when you want to get a sense of what your deep ancestry is, which is not where did my grandmother come from, but 30,000 years ago, 15,000 years ago, where did my ancestors come from? They look at changes that are common to your genetic information that tell us about your ancestry. This is used increasingly for disease risk to identify diseases that you are at a higher or a lower risk for, well before you develop symptoms. That's something called personalized medicine that we're going to dig into in just a second and help shape treatment. I want to make a distinction between single gene disorders, which are probably the genetic disorders you're most familiar with, and complex. Let me do this quickly. Single gene disorders are caused by a change in a single recipe. Sickle cell anemia, cystic fibrosis, many forms of achondroplasia, which is short-limbed dwarfism. Many of the things like polycystic kidney disease or fetal ketonuria, PKU, many of the things that they prick an infant's heel for at birth and look at the blood, these are diseases caused by one change in a single gene, sometimes 2 changes in a single gene. But it's a single gene, and when you change that gene, you lead to the development of the disease. And they often show up in these kinds of patterns, which I bet you are having bad flashbacks, or maybe you're having good flashbacks from biology class. When you talked about Mendel's peas, and tall and short, and green pea and wrinkled and non-wrinkled, They're caused by changes in a single gene, and you can see them move through families in very specific patterns. There are several thousand single gene diseases that affect humanity. Individually, they are rare. Collectively, they're quite common. Much work in genetics has focused on identifying the causes, the changes in DNA that lead to single gene diseases. Let me make a switch now. Let me talk about complex. Most of the common traits, most forms of dementia, most forms of cardiovascular disease, most forms of diabetes, most forms of cancer, most forms of hypertension are complex diseases. They're not caused by a change in a single gene, but instead they're caused by multiple changes across several different genes plus environmental factors working together. So think about diabetes, for example. Individuals that have diabetes, whether we're talking about type 1 diabetes or type 2 diabetes, probably got the disease because they had changes in somewhere between 5 and 15 different genes, each of which increased their risk just a little. And it was the collective impact of these changes in these multiple genes plus being in a risky environment. And what I mean by risky environment isn't that they were doing stupid things, but they had a genetic predisposition because they had these risk versions, and they were in an environment that also increased the risk. Now, do we know what those risk factors are? Unfortunately not. Otherwise, we'd know how to prevent them. But it isn't enough to just have the genetic risks. You also have to have the environmental risks, and it's those pieces working together. That is what we are putting enormous amounts of energy here at HudsonAlpha and really in research around the world to try to better understand. Because if all of you in the room, let's say you're all impacted with type 2 diabetes, unfortunately, you're not all going to have the same set of genetic and environmental risks. There may be 40 different potential combinations, and you may have come to diabetes, type 2 diabetes, by different blends of genetic and environment. This makes it really, really difficult to identify the genetic changes and the environmental changes because the population is so mixed. Does that make sense? It's very difficult to get at. We're beginning to have tools that let us get there, but it's very difficult to get to. We can continue the analogy of how genes are like cake recipes by thinking of your genome as being like a cookbook. The genome contains all the genes that an organism needs to carry out life in the same way that a cookbook contains all specific set of recipes. Interestingly though, at least in the human genome, a tiny fraction is actually these genetic recipes. The rest of it, over 99%, is probably regulatory, although we're still probing the mysteries of really understanding what that means. Thanks for watching. I hope you've enjoyed this segment.